tissue culture. A culturing of tissue or growing of tissue or one organ or single cell also. We can grow the here plant cell. Plant cell generally having the totipotency character. So, any plant cell from root or stem or leaf or flower from any the cell from here any organ in the plant, any cell can produce one total plant. It is having the plant cell is having the totipotency character. Totipotency nature is present in only plant cells. It is a very special feature of plant cell. And here any living cell, any living plant cell can develop into it is plant cell. The plant cell when it is grown on the medium or the suitable conditions, here in the suitable environmental conditions, here if the plant cell is grown on the medium, that develops into a new plant, total plant. The total plant we can develop from a single cell or from tissue also or from some group of cells also. Here the totipotency character is observed in only plant cells. It is the special feature of plant cell. Next here how the plant cells are grown on the medium. So, here generally for the plant growth here different atmospheric conditions and some nutrients are also here essential for the growth of plant. For the germination of the seed and for the growth and development here some requirements are needed. So, these requirements are light, water and nutrients and some here temperature. So, different environmental conditions, here environmental conditions and raw materials. So, all these are needed. Suppose here the total environmental conditions, temperature, light, pressure and soil is generally it is giving the anchorage. So, like here the medium is it is giving the anchorage to the root system. So, here plant generally it grows in the soil. So, from the root system is absorbing the water from the soil and nutrients also, and it is synthesizing the food materials from the carbon dioxide and water during the photosynthesis process. So, here all the enzymes and hormones, so all they are synthesized within the plant body, but here we are providing. So, all these enzymes, hormones and nutrients, water, so all we are providing to the plant cell. So, when all these are provided, so the plant cell develops into a total plant. Here the plant cell generally when it is grown on the nutrient medium, that is developing into the total plant. So, what is nutrient medium? So, how can we prepare the nutrient medium? So, let us discuss. Preparation of explant. So, what is explant? So, explant is taken from any part of the plant. So, the explant is here the group of cells or some tissue or some part of plant that is taken from leaf or flower or anther, from any living part it is taken. So, for example, it is one leaf. Here some tissue is it is taken and that is cultured. The tissue from the leaf, it is cultured in the nutrient medium. So, here this small tissue is called as it is explant. So, explant is a group of cells or tissue and that is cultured into the new plant. So, first preparation of explant, it is the first step. So, after the preparation of explant, it is sterilized. It is having number of microorganisms also. On this microorganisms are also present. That is why before culturing the explant on the medium, it is sterilized. It is sterilized in the alcohol. So, the tissue is sterilized in the alcohol. So, otherwise it is completely spoiled by microorganisms on the medium. So, it is the first step, preparation of explant. Second step is preparation of nutrient medium. The nutrient medium, it is having all the enzymes, hormones and nutrients or minerals. So, water content, carbohydrates all are present in the medium. So, here the nutrient medium during the preparation, a preparation of nutrient medium. So, nutrient medium is containing all the requirements here. The sterilized water, so pure water, sterilized water and the agar is water and agar generally it is obtained from the rhodophycean algal members like Gelidium, Gracilaria, Zygotina from the cell walls or from the algal members. Here the agar it is a carbohydrate, it is obtained, and this agar is used in the preparation of a nutrient medium. So, generally here the different media are there, but here generally used medium is MS medium. MS medium. MS medium. So, it is the scientist, the M means Murashige and Skoog. The two scientists developed this medium. So, this medium contains water, agar and here hormones, plant growth regulators like auxins, cytokinins and gibberellins like this. All these are plant growth regulators. Mainly here auxins and cytokinins are used, and some nutrients are also hormones and enzymes and some nutrients, micronutrients or minerals. So, all these are mixed. So, that develops into that that is the medium is prepared. The medium is generally here MS medium, Murashige Skoog medium. Murashige Skoog medium is generally used to culture the tissue or cell or one organ in the tissue culture technique. Here it is a medium. The medium is poured into the vessels. So, it is test tube or in conical flask. So, generally that is taken into the test tube and it is poured. After some time here it is solidified. For the solidification here the agar is used. So, it is like a semi solid like structure, semi solid material like gel material. So, it is not completely liquid, some liquid material, liquid nutrient media are also there, but here it is solidified, the water and all these components and agar when the agar is when the agar is used, so it is solidified. It is a semi solid like material, and here the surface area, on this surface area the explant, this explant is yes it is inoculated, the explant is inoculated onto the medium. So, before the inoculation of explant onto the medium, so both are sterilized. Sterilization means the removal of microorganisms from the medium or from the explant also. Suppose here without before the sterilization, this material or this explant is cultured or it is cultured on the medium, when it is transferred onto the medium, so that is having number of microorganisms. So, here the microorganisms easily or very fast, rapidly they can develop on this medium. Why? Because all the here food materials, sufficient food materials are there in the medium. That is why microorganisms easily develop on the medium. So, here before the inoculation, this explant and medium completely they are sterilized. So, the second here third step is inoculation step. Inoculation. So, inoculation is nothing but the transfer of explant onto the medium. So, it is completely carried out in aseptic conditions. Aseptic condition means a microbial free environment. So, where the microbe in the lab generally the microbes microbial free environment the explant is transferred onto the medium. So, after the transferring the explant it is developing into the tissue or group of cells or next embryo like structure. So, the after the inoculation it is incubated. The fourth step is incubation. Here incubation, after transferring the explant onto the nutrient medium, it is incubated. Incubation means under suitable conditions, temperature, light and environmental conditions. Here the explant is incubated, here explant is inoculated. After the inoculation it is incubated. So, incubation is carried out in here sterilization. Third step is sterilization. Here after the preparation of explant and after the preparation of here this medium completely, the media is having number of here materials, suitable food materials and hormones, enzymes. That is why the microorganisms easily grow in this medium, and explant also to be sterilized. So, before the inoculation all the explant and media both are sterilized. Sterilization means removal of microorganisms from the medium or from the here explant, and so many equipments are used in the here in the tissue culture. So, the tissue culture, during the tissue culture the equipments are also here sterilized. So, the sterilization is carried out under 121 degrees centigrade for 121 degrees centigrade and 15 lbs pressure for 15 minutes. So, the total vessels which are used in the tissue culture technique, all these are here sterilized in the autoclave. So, autoclave is a pressure cooker like apparatus. Here high temperature 121 degrees centigrade temperature and high pressure is also present, 15 lbs pressure. For 15 minutes it is sterilized. After the sterilization, here the medium generally the media and vessels are here sterilized. Here the total microorganism, if any microorganism is microorganisms are present in the medium or in the vessels, they are completely killed by this process. But here the explant is not sterilized by this method. Why? Because here the explant is a living tissue. The living tissue is sterilized with the help of a small quantity of alcohol. So, the explant is sterilized by different method, but here vessels and media are sterilized by here high pressure and 15 lbs pressure, and for 15 minutes it is autoclaved. So, autoclave is a pressure cooker like here equipment, and here it is after the sterilization process the explant is inoculated. So, the fourth step is inoculation. Inoculation. So, the insertion of the explant onto the medium, it is called as inoculation. So, it is carried out under suitable environmental conditions or in aseptic conditions, where the microbial free environment is there, that is aseptic condition. In aseptic conditions, here the inoculation is carried out, and after the inoculation it is incubated. Incubation means the medium and the tissue, they are allowed to grow. Here the generally the tissue develops into a new plant. So, during this development some group of tissue or embryo like structures are formed. So, the incubation under suitable environmental conditions the tissue is allowed to grow. So, in the suitable environmental conditions. So, after the inoculation it is completely covered with a cotton plug. It is a cotton plug. So, the cotton plug, why the cotton plug is used? Here the cotton generally it is allowing the exchange of gases, oxygen, carbon dioxide entry and exit. So, from the atmosphere the oxygen, carbon dioxide here can exchange through the cotton plug, but microorganisms, microbial organisms they cannot enter into the into this vessel or into the test tube. So, it is allowing the exchange of gases, but it is preventing the entry of bacteria or microorganisms. So, if the microorganisms enter, so before this explant they will grow fast, where they will develop into the, they will develop and completely the tissue culture explant it is spoiled or damaged. That is why here after the inoculation and after the inoculation it is incubated. So, before the incubation here it is covered with a cotton plug. So, gases exchange is taking place. Next, after some time, means here 3 to 4 weeks. After 3 to 4 weeks, so this tissue develops into, here one cell divides into 2 cells, 4 cells, like this number of cells. So, group of cells are formed. So, this group of cells is called as callus. Callus. So, it is the undifferentiated mass of cells. So, group of cells that is called as callus. Callus generally it is developing into here one embryo like structure, and sometimes it is also directly developing into the here plant, new plant. So, the callus is developing into embryo like structure. So, the embryo like structure is called as somatic embryos. Somatic embryos or embryoids. So, this callus is undifferentiated mass of cells, and it is developing into one embryo like structure. So, the embryo like structure, embryo generally it is the result of sexual reproduction. In the sexual reproduction zygote develops into here embryo, but here this embryo like structure is not formed from the zygote. That is why here it is a somatic. Somatic means vegetative. From vegetative cell, the embryo like structure is developed. So, somatic embryos, also known as embryoids. Embryoids. It is also called as somatic embryogenesis. Somatic embryogenesis. So, callus is the mass of undifferentiated group of cells, and that is developing into embryo like structure. And this embryo like structure is called as a somatic embryo or embryoids. This embryoid or somatic embryo develops into new plant. So, that new plant development is depending on the auxin and cytokinin ratio. So, here first which is developing a root system or shoot system. The root system and shoot system development is depending on the auxin and cytokinins phytohormones here concentration or ratio. Here the embryoid generally it is developing into here organs, or organs are formed first. The organs are a root system and shoot system. Here the root formation is called as rhizogenesis, and shoot formation is called as callogenesis.